Hello everyone, today, it's my pleasure to introduce Wikomo Bioin Cloud, a user-friendly platform for meta-omics data analyses. The concept that cancer is a systemic disease involving neural regulation is now widely recognized. 1. Continuous advances in molecular biology have significantly propelled oncological research. Oncology has shifted focus from intratumoral mechanisms to the tumor microenvironment. 2. Importantly, viewing tumors as self-regulating pseudo-organs rather than tissues to investigate their systemic effects offers a novel perspective on tumor biology. 1. Currently, research on intratumoral neural infiltration primarily focuses on molecular mechanisms, which holds advantages. However, a more macroscopic and holistic research perspective is needed for neural innervation. 3. We propose that organs serve as the fundamental biological units for understanding cancer neuroscience. Figure 1. Organ is the basic unit of tumor nerve research because of the organ heterogeneity of tumor innervation. 4. 5. Central neoplasms such as glioblastoma instead hijack existing neurons, forming functional synapses that drive activity-dependent proliferation. 6. While peripheral neoplasms, pancreas, breast, etc., recruit mainly peripheral autonomic, sympathetic/parasympathetic, and sensory nerves via axonogenesis and perineural invasion foster a pro-tumor milieu. Visceral tumors, liver, colorectum, receive dual autonomic supply and exploit metabolic neural coupling, e.g. vagal sensing of portalosmolarity, or cholinergic/adrenergic imbalance to propel growth. 7. Somatic tumors, skin, soft tissue sarcoma, display segmental somatic sensory innervation, TRPV1+/CGRP+ fibers, conveying sharp pain and mechanosensation, with scarce autonomic input. 8. As tumor immunology has evolved from the generalized immune evasion paradigm to a framework of organ-specific immune environments, an organ-based approach is similarly beneficial for comprehending and overcoming cancer. Organ-specific neural infiltration and neural innervation. Neural infiltration and neural innervation represent distinct states within tumors. Compared to adjacent normal tissue, solid tumors exhibit significant hyperinnervation, characterized by increased nerve fiber density, aberrant branching patterns, and dysfunction. 9. 10. This process is termed axonogenesis or sprouting, involves chemotactic attraction of adjacent nerve terminals by tumor-secreted neurotrophic factors, e.g. NGF, BDNF, artemin, and axon guidance molecules, e.g. netrin-1, ephrin B1, inducing axonal growth from dorsal root ganglia, DRG, and autonomic ganglia into the tumor parenchyma. 6. 11. Different organs exhibit distinct peripheral neural innervation backgrounds. 4. 5. Visceral organs, pancreas, prostate, lung, gallbladder, receive dense dual autonomic innervation, sympathetic adrenergic fibers and parasympathetic cholinergic fibers, along with sensory nerve fibers, whereas somatic tissues, skin, breast, oral mucosa, are primarily innervat by sensory nerves with sparse autonomic distribution. 4. These distinct neural backgrounds give rise to different dependencies. Prostate and pancreatic adenocarcinomas exhibit autonomic addiction, with sympathetic nerves driving early tumorigenesis via beta-adrenergic signaling and parasympathetic nerves promoting metastatic dissemination through muscarinic receptors. 12. In contrast, cutaneous malignancies, melanoma, basal cell carcinoma, and breast cancer rely primarily on sensory neuron-derived neuropeptides, substance P, CGRP, for growth and invasion. 5. Crucially, the developmental origin of tumors dictates their neurointeractome. 13. Organs of endodermal origin, pancreas, biliary tract, and mesodermal urogenital tissues retain the plasticity to recruit autonomic precursor cells, while tumors of ectodermal origin hijack existing sensory neural structures. 14. Studies have found that the degree of neural infiltration significantly correlates with lymph node metastasis and organ